the director of the NIH, Francis Collins. We're very glad to have with us today. Francis, in addition to being the director of the NIH, is an eminent human geneticist who was instrumental in the Human Genome Project and remains involved. He's an outspoken supporter of the NIH mission to support a diverse portfolio of research projects, including the pursuit of fundamental biological knowledge. And he's here to discuss accelerating insights from basic genetics. Thank you for being here, Francis.
Thanks, and good morning to all of you. It's really a privilege and an honor to be part of this remarkable plenary session. Three wonderful talks have preceded me, and believe me, I wish I could give you a presentation on stretch enhancers or on mouse models of progeria, things that are going on in my own lab. But I was asked instead to sort of step back and offer some view from the perspective of the NIH director on the support for basic science and particularly support for model organisms, something that is near and dear to my heart and to that of many of the other leaders at NIH. So I want to present you not just with some exhortations and reassurances, but also with some data to show what we're actually able to do these days in terms of support for the work that you all do. And I think much of this will be reassuring. I also want to provide some exhortations, though, about ways that we might be able to get the research on model organisms even more effectively integrated across different species, as this whole meeting has basically exemplified and has, I think, been the intention of the organizers. How can we actually take this to the next level in the 21st century, where we can learn not just from one model organism at a time, but across multiple organisms simultaneously? And how can we empower people to do that? Because many researchers are in fact interested and are already taking that tack. But before I go any further, though, I do want to make one very personal recognition, and that is in memory of a dear friend, Bill Gelbart, who had such a powerful influence on many of us in terms of emphasizing the things that one could do with model organisms, particularly in his case, Drosophila, and the way in which it was possible to empower others by creating databases, namely Flybase, and that are used by so many people so productively. And Bill was also a wonderful advisor to all of us working on the Genome Project. He ultimately became the chair of the sequencing advisory panel that I put together to oversee what we were doing with large-scale sequencing. A real hero and someone that all of us who knew him now miss very much. So this is in memory of him. Well, I think we should also recognize this is a special year, and that's one of the things that's being celebrated here, the 100th anniversary of genetics. And if you've looked at the evidence of what was in that first issue, it was pretty interesting, non-disjunction of proof of the chromosome theory of heredity. So we've come a long way in a century, but we have a long way to go if we were really going to say we fully understand all of these organisms. But it could hardly be a more exciting time. And for the many young scientists that I see in this gathering, you've arrived at just the right moment in order to be part of a major revolution in our understanding of biology, which is going on all around you at this meeting and will only accelerate as we go forward in time. Now, NIH's role in all of this is significant as the largest supporter of biomedical research in the world. And we have a dual commitment to both the basic and the applied part of that. Our mission statement here specifically talks about fundamental knowledge about the nature and behavior of living systems, but also about the application. And people often ask, well, what's the balance there between basic and applied? And is there a shift that's been going on over time? Perhaps there's a sense that, in fact, NIH is now more pushing towards translation and not paying as much attention to basic science as we used to. Actually, the data doesn't support that, although the stresses that all of us have been feeling for the last 13 or 14 years with declining purchasing power for research may make it feel as if your particular area is under more stress than the rest. But actually, we're all pretty much under stress, and if you look at the evidence, it looks as if the balance has been fairly constant. And I hope I can convince you that from the leadership of NIH, the support of basic science, not attached to any particular disease or any particular therapeutic consequence, is strong and remains so and has been in that space over the course of many decades. Here's a quote from Jim Shannon, the 8th director of NIH. This is from long about 1960. We're talking about the importance of basic science. As NIH director, I had a chance to write an editorial in Science magazine back in 2012, also talking about how critical it is for us to continue to focus on basic science, because almost everything that we've learned about biology has started in that space. And more recently, just earlier this year in Science magazine, in a brief letter that was co-signed by every single one of the NIH Institute and Center directors and office directors and senior staff in my office, basically made this point all over again, that science is a bedrock of progress. The taxpayer investment in NIH has yielded spectacular returns, and we must continue our vigorous support of this. So I hope those of you who saw that were reassured that there is no, I repeat, no tendency at all to shrink back from NIH's responsibility and enthusiasm for the support of basic science. If you look at the statistics, oh, by the way, it pays off pretty well here if you count Nobel laureates. Most of those have, in fact, been in the area of basic science. Here's what you can see if you basically look across the entire NIH portfolio for what we would call basic and what we would call applied. We can quibble about how those definitions are determined, but we use the same definitions over many years, so you can at least see if there's a trend. And while you might see there's a slight diminution of basic science support, boy, it's pretty small here, with about 52% of our portfolio in the basic and the remainder in applied. And that, of course, varies from institute to institute, with some institutes like NIGMS focusing very heavily on basic science, and others like, say, NCATS, the National Center for Advancing Translational Sciences, by their very definition, having a more applied focus. But this is what you see across the entire portfolio. And I would like to say that from my perspective, the delivery of exciting results coming out of the model organism community, represented so nicely here at this remarkable gathering, is phenomenal. And these are just a few randomly chosen publications coming out of your own areas of research. And that's something that I have a chance as the NIH director also to write about. Every Tuesday and every Thursday, I put out a blog, and I hope you're all reading it. And I get to pick something that's just appeared in the literature that I think is particularly interesting and try to explain it to a broader audience. And just over the course of the last year or so, here are some examples of the blog that focused particularly on model organism developments, because there was lots to pick from there. All of these fields are vigorously moving forward. But I think people are wondering, okay, that sounds good, but how are we actually doing as far as the support of model organism research? Some of you probably saw this article that was published a little more than a year ago by Wangler, Yamamoto, and Bellin, which basically tried to determine, is there in fact some diminution in support for Drosophila research? And what they did was to look across the NIH portfolio, because we have that out there in the database where you can look at it, and scanned through to see grants where the word Drosophila appeared either in the title or the abstract, and look to see what the trend was. And it was somewhat disturbing, because as you can see from the red bars here, the suggestion was that there had been a drop-off beginning in about 2009 in the number of such grants that were funded by NIH. And that might suggest that there is some gradual discrimination going on in the peer review system in terms of support of this sort of research. So we were concerned about that too. I must tell you that we now have at NIH a variety of tools to be able to do very sophisticated portfolio analysis, including a whole office now that has brought many of those tools on board. So we can go beyond a simple scan for keywords in titles and abstracts. And I asked my team to look at that and see if we could, in fact, try to verify or perhaps come up with a different answer to this, because this did not seem consistent with what many of the people that I talked to thought was the case. And here's what we found. This is now actually plotting not numbers of grants. This is dollars invested. But if I plotted numbers of grants, it would show a very similar trend. So pay close attention here to the various bars. The yellow line there is basically the millions of dollars, of R01 funding for across all of NIH. And you can see what's happened there. Notice, by the way, 2013, that drop, that was sequester, and sequester is unfortunately still a threat to us. And then you can see in terms of what the Wangler analysis that I showed you a minute ago is the lower bars here, which basically was able to capture those grants where Drosophila specifically appeared in the title or the abstract. Our analysis, however, was able to look at the specific aims. And by the way, we did this in two different ways. One was by a completely automated process, and the other was manual curation. So I think these data are very dependable. And I think what you can see, and I hope you'll be reassured by, is that in fact, the simple analysis misses a lot of grants, and the evidence here that there's actually been a deterioration specifically of Drosophila dollar support doesn't seem to be borne out once you have all of the data. You can see a little drop-off in 2013, but that was a drop-off for the entire portfolio. You may be wondering, well, what about success rates? And I'll come to that in a bit. But first, let me show you what's happened with the other model organisms that are so ably represented here. By the way, the one I can't show you is yeast, because we found it very difficult to capture, even in these various text mining ways, which grants are involved in yeast, because any grant that mentioned a yeast-2 hybrid somewhere in the text would get called a positive, and obviously that's probably not what we're looking for. We are working on the Saccharomyces, so apologies to the yeast folks that I can't show you that data, but I'll show you some others. So C. elegans, for instance, now I'm plotting not dollars, but I'm plotting numbers of R01 grants. And this is both new and competing and non-competing. So you can see all R01s, the yellow line here, is in the range of 25,000 or so. And C. elegans R01s have actually been following a pretty similar trend after a pretty significant increase going from 2009 to 2012, and now tracking along somewhat similar, perhaps slightly dropping off, but not much more than the rest of the grant portfolio. By the way, it is troubling, and it should be to all of us, that this number, the yellow bar, is actually not going up at the present time, but that's a consequence of what's happened to overall NIH funding, which I'll come to at the end. Zebrafish. Zebrafish is actually doing quite well here. You can see the number of R01s is going up on a curve that is in the upward direction, whereas the rest of NIH is slightly trickling downward. So clearly, this is an area of lots of scientific energy and activity. Mouse turns out to be a bit challenging to figure out what is a mouse grant. So what we did in this case was to scan through and see, did the grant in its animal use section mention that mice were being involved in that particular proposal? It's interesting, in this case, I'm just plotting new and competing R01s, not the non-competing. That's why the numbers are much less here as far as all R01s. But notice that the scale on the right and the scale on the left are just a factor of 2 different. That is saying that almost half of NIH-funded new and competing R01s are using mice, which is pretty astounding. That's maybe a bit higher than I would have guessed, but there you have it. And you can see the curves are tracking quite much in parallel, perhaps in fact because we're talking about almost half the portfolio. Now, what about success rates? Pay close attention to this, because this was maybe the thing that I was most concerned about, pulling out this data and trying to analyze it. Is there something going on here where model organism grants, when they come in, are not faring as well as they should in peer review because there is some bias happening? It looks as if anything, it's the other way around. So again, look at the figures. In each case, the yellow bar, yellow line, is the success rate from 2008 to 2015 for all R01s. And it is a great source of distress to me and to all of you that is below 20% and has been so, when it traditionally has been in the space of 30%. So much of the pain we have all been feeling is represented by that yellow line. But if you look at success rates for Drosophila, C. elegans, zebrafish, those, with almost no exceptions, are substantially higher than for the rest of R01. So if you're sending in a grant with these particular model organisms as your main focus, your opportunity to get funded is better than somebody who's doing something else. The mouse is almost identical, again, because we're talking about half the portfolio. Perhaps that's not too surprising, but there's certainly no discrimination happening against the mouse. And for the other organisms, again, I hope you can see here, things are looking awfully good. So I would say to all of you, if you want to see more research done on model organisms, send in more grants. They're doing pretty well here. And it looks as if that trend has been continuing over multiple years. I hope that is reassuring to people who have wondered, in this difficult time where everything is getting squeezed, is there a particularly severe squeeze happening to model organisms? The data here would indicate that, in fact, that is not the case. If you're interested in looking at the more detailed analysis that I'm just giving you a snapshot of, this particular blog written by Mike Lauer, who is the Deputy Director for Extramural Research and who posts all of his information on a blog called Open Mike. This was just posted probably minutes ago, so that it's up there if you want to look at even more figures, even more data. This is very much the way Mike has approached many of the questions about what we're doing with our grant portfolio. Let's get the data and let's see what it shows. Now, let me talk about data in other ways, because I know this is a topic also of great interest and perhaps some concern in this audience. What are we doing as far as the model organism databases, and how can we be sure that that critical part of the vigor of your communities continues to be the source of the kind of information you're looking for, and even gets better as we go forward in time, as more and more data gets generated and there's more and more interest in being able to compute across model organisms and not simply within a single organism. This is just a snapshot of an area that I can tell you is taking a great deal of the time and energy of all of us at NIH, trying to figure out what to do with the era of big data, which has really arrived. Phil Bourne, who's my new director of data science, made an estimate of what NIH's production of data was last year, and came up with a reasonable but probably rough estimate of 650 petabytes. Now, maybe that doesn't sound like a lot, but actually, when you consider that the entire content of the Library of Congress is about 3 petabytes, that tells you what we are doing here in biological science in terms of the incredible volume of information that's getting produced. And of course, that's not a static number, that's going up rapidly. We have replaced cosmology and satellite weather images as the place of the most rapid growth in data, and we have to catch up quickly with what might have been not the most vigorous response to that opportunity over the past few years. And that's why at NIH we are spending a lot of time and a lot of money trying to figure out how best to manage this, so that data is captured, it is placed somewhere where you can find it and you can compute on it, it is well marked with meta tags and so on. And without a lot of community involvement and support for this, we're going to drown in our data as opposed to benefit from it. And it would be really tragic if we missed this opportunity. So what are the things NIH has been doing? 4 years ago, I asked my advisory committee to set up a working group to ask what should we be accomplishing that we are not, and they produced a very vigorous set of recommendations. One of the recommendations says, if you don't basically act upon what we're telling you, you're committing institutional malpractice. So they were pretty strong in their view that we had a lot of work to do. That led to the launch of a program called BD2K, Big Data to Knowledge, which is now being ramped up to about $100 million a year, basically with many different tasks, but mostly supporting research and training. And we formed a scientific data council at NIH within that to try to manage the scientific priority setting, because this crosses many different institutes. It can't live within just one. I did hire Phil Bourne from UCSD as my first associate director for data science to oversee this issue across all of the institutes. And then we also looked at the National Library of Medicine as a place where there should be lots of focus on data science, and formed a group to look at what the future of NLM might need to be, and just recently appointed a new director, Patricia Brennan, as the director, who will be arriving in about a month. The BD2K program, which is originally gotten started in the director's office, is going to migrate to NLM, which means that will become a very important place for all of our data science activities and discussions to sort of have a hub of activity. And very recently, in the last couple of months, I've created a trans-NIH data infrastructure task force to wrestle with the question about where is this data going to go. We all understand that ultimately, if this is going to be accessible, it needs to be in the cloud. How do we manage that when we consider all of the things that are already existing on various servers whose future lifetime may not be all that well defined? So many things going on in this space. What about model organisms? Well, obviously, I would say model organisms are a great example of really excellent data management, but things are evolving, and I would like to see model organism databases also out there in front in terms of evolving into this next era of cloud management and of access and availability for researchers who have creative ideas that you might not have estimated were possible a couple of years ago. Efforts to manage the data in model organisms have certainly been around for a while, going back from some of the early documents you see here. And then with the Genome Project and the generation of reference genomes on these organisms, model organism databases emerged, very nicely curated, and became of great utility to the communities as they are today. And I think the signs of success there are remarkable. Data has expanded beyond genomics. Lots of researchers use these tens of thousands every month. And NIH funding has, over the course of many years, been significant and has remained steady, as you can see in this graph here. Most of the dollars, what's in yellow, have been coming from the National Human Genome Research Institute, with only small contributions from other institutes. There's a little blip there in 2015, which was a contribution from the Data Science Office. This is a total of the annual funding of the 5 model organism databases and the GO consortium. But as you may know, there's now some discussion about where this should go. It's not a discussion that's necessarily brand new. Here is a statement about the need for coming up with ways to make the model organism databases more integratable. For the most part, they evolved independently with a lack of standards for developing, curating, and maintaining these databases. Potential exists for incompatibility and inconsistencies. A pressing issue is to develop methods to make them interoperable, to come up with standards. When was this statement made? Actually, 2002. So it's been true for a long time that we are appreciating an opportunity and a need to make the model organism databases take on more of this set of new opportunities. But that's been challenging to do. Each of those have been operated, I think, extremely effectively by a principal investigator, by a very invested community, and the idea of change isn't always an easy one for people to adopt. But I think the change has now sort of reached the point where we really have all the more reason to do so, and NHGRI has certainly been pushing the idea that this no longer is something to keep talking about. Let's see what we can do. The databases that NHGRI is proposing some reorganization are listed here. The goal to improve the usability of these for the wider community, to preserve the value in the services provided, but help also to have a more sustainable funding model commensurate with the scale and scope. And that would mean adopting some updated management practices, improving integration of content, enhancing interoperability, do more exchanging of best practices between the model organisms. I haven't talked to too many people who say, oh, well, you shouldn't do that. This all just makes sense. I think the concern is, along the way, will there be any degradation of the function of the databases that each model organism has grown to depend upon, the community has. And we are also very concerned that that not happen. But we do have to figure out how to induce these steps forward, and some change is going to definitely be needed. One of the changes that needs to happen, and which is also a great opportunity, is to move the data into the cloud. We can't afford, with the way in which all of the data is being gathered, to be able to maintain it on individual servers. And the cloud provides all sorts of opportunities also in terms of ease of manipulation, where you don't have to send very large data sets around in very slow downloads. And BD2K is pushing very hard on an initiative which ultimately we aim to put most of the biomedical research data that NIH supports into cloud-based databases. And this would be called a commons, a virtual ecosystem where scientists can work with the digital objects of research, but all very nicely indexed so you can find them according to the FAIR principles. And that is also, I think, a circumstance that needs not to be just introduced in one fell swoop. There need to be pilots to see how the commons could work. And there's a general sense that there's some very attractive pilots, and the model organism databases are one of those. And that kind of effort is already being supported, and already your leaders of the model organism databases have submitted administrative supplements to see how that particular process could begin of moving the model organism databases into the cloud. Not in a way that should make it harder for people to work with, but to make it easier instead. So there are, of course, other things going on in this space in terms of what we are trying to do about the commons and dealing with data science. The model organism databases is one part of a larger discussion. If you haven't seen this particular outlook in Nature, you might have a look at what Phil Bourne and John Lorsch and Eric Green have written about this whole idea about where we need to go with biomedical data. I don't think it's appropriate anymore for people to kind of assume that's somebody else's problem. This is going to be so central to our communities going forward that we all have to kind of become data scientists, or we will suffer the consequences. In order to try to see how these steps might be taken forward, the model organism database PIs were convened by NHGRI in May 2015, and then again a couple of months ago. Idea exchanges were put forward and administrative supplements solicited to begin integration. As you can see, there is in fact now the Alliance of Genome Resources, and I saw they have a table here at the meeting about how this can be achieved, recognizing that there's work to be done here, but it's time to get underway, and I'm really glad to see this being now embraced by the leadership of those databases. There's also a discussion amongst many other potential funding sources about how we might do this. The Genome Institute, because it is modest in size, is not quite clear about why the dollar sources for this should be coming exclusively from just one part of NIH, and would like to see this more broadly supported, and we're certainly figuring out some possible ways to do that. A letter has been written, as many of you know, addressed to me and the NIH Institute directors. I think a very thoughtful letter, basically endorsing in a general way the need to come up with a more integrated set of model organism databases to enable better research, but also expressing concern about not moving so quickly with reductions in the overall support of the individual databases that function would be degraded. I've read that letter carefully. I understand that, at least as of yesterday, there were more than 10,000 signatures, so that does tend to get one's attention. And I want to reassure you that all of us are taking this with great seriousness. So we are hearing the concerns. Believe me, I am hearing them from lots of you. But I also want to emphasize, just to continue down the path we are now with the separate model organisms and not figuring out how to get them to talk to each other, that's not the right answer either. So status quo is not an option. Talking to Eric Green, he has now agreed that we need to have another deep conversation about this before figuring out exactly what this trajectory ought to be. So there will be convened by NHGRI another meeting of model organism PIs and community members included in that this fall, and Eric would appreciate input from all of you about who the appropriate community members might be in a meeting that can't be too huge, but ought to represent the concerns and the perspectives of people who really understand these issues. So that will be going forward, and I don't want anybody to become alarmed that some tempestuous changes are going to happen in this space until we've had an opportunity for another conversation about what this path needs to be. But the path does have to involve change. So I think where we want to get to, and this will probably not come as a surprise, is a single federated view of the model organism database that you could actually use in ways that are currently quite clunky to try to anticipate how to get answers from. At this plenary session, we've heard from all of the speakers about how model organisms inform each other and how they inform human biology. And yet, some of the questions you'd like to ask that involve multiple model organisms are at the present time not so easy to do. Just a simple question. You want to search for human orthologs across multiple model organisms. Today, you've got to query each database separately. You've got to download the data, then you've got to compare it. It turns out the orthologs are calculated differently. The data is stored differently. It seems like a pretty fundamental question that you ought to be able, with all of the smarts and all the data that we've got, answer that without having to go through the steps that we now do. So this kind of query, it seems to me, is the sort of thing as a use case that one ought to imagine coming up with a better way to take this powerful set of resources and make them even better. And I would like to say as an exhortation to all of you and to the database PIs and all the people involved in the sort of data science of model organisms, you guys have been out in front for a long time in this idea of how you take public data and you make it more useful. Why don't you continue? Lead that big data revolution in biomedicine by figuring out how to do this integrating part as well in the spirit of the cooperation that guided the Genome Project and model organism research. Finally, before I let you all go off to the next set of remarkable gatherings at this meeting, I want to finish, though, with just one mention of the state of biomedical research funding, both by way of information, but also by asking for assistance from all of you to make the case for the value of biomedical research in every way that you have a chance to do, because it's critical right now. I think we would all agree, and this meeting represents that we are in exceptional times scientifically, but we are also in exceptional times as far as the financial stress that we've been under since 2003. This diagram shows really in graphical terms why things have been tough since then. That white line is essentially the purchasing power of NIH to support your grants and those all over the country and some outside the country. So it's corrected for inflation. You can see the nice doubling that happened between 1998 and 2003, but then you can see what happened after that. With flat budgets, inflation eroding purchasing power, the sequester that came along right there and cost us $1.5 billion in one fell swoop, from which we've still not quite recovered. By the way, that dotted yellow line is what NIH trajectory would have been if we'd stayed on the same path we had been since 1970 with basically inflation plus 3.7%. We would have been better off by now by a long shot. But I'm not saying the doubling was a bad thing. It's just once the doubling happened, there was this sense, oh, NIH's been taken care of. And then we became undoubled over the course of the next dozen years. This is why things are so tight all around, and this is why when you look at grant success rates, which I showed you a small part of the more recent years, this is what it looks back if you go to 1978, you can see that until 2003, we were bouncing around in that space around 25 to 30%, and we are now below 20. We need to get back into that positive zone. I can say with some optimism that this year, FY16, for the first time, Congress came through in a very significant way with a $2 billion increase for NIH, and that was a bipartisan effort. It was a result of a lot of leadership from the administration and from both the Senate and the House leadership managing appropriations, and I hope everybody sent them love notes at that point because they deserve them. And we are now right in the crux of trying to figure out what's going to happen in FY17. Both the Senate and the House have come forward with tentative marks that include an uptick, $2 billion from the Senate, $1.25 from the House. Again, we should be very grateful to those folks for coming up with this kind of proposal at a very difficult budgetary time. But the future is very uncertain. All I'm saying is we should, in fact, all of us, not be shy to talk about why it is that science is both such a remarkable human endeavor, but it is also a way in which we advance the cause of medicine, and it's also the way in which we stimulate the economy, which happens to be true as well. And I hope if you've not invited your elected officials to come and visit your lab and show them what you do recently, that you might consider doing that. That's the single most effective thing that is available to us in telling our story. And finally, I would say we do need to do this all together. I'm glad to be here with all of you, sort of back with my peeps here. It feels like having worked in model organisms and in biomedical research, and particularly in genetics for my whole career. But if we can, in fact, figure out how to tell our story and to tell it effectively and well, it will be much better to do so as a group. One voice is really good. Put it all together, it makes a much better musical contribution. And I hope our symphony of voices about the excitement we all feel and the potential it has for changing the world will come across loud and clear. Sometimes it feels like we're going awfully fast. We are. Sometimes, maybe as with the model organism databases, it seems as if NIH is pushing you to make changes too quickly, and I can understand that. I'll just finish with a wonderful quote I just heard, which is attributed to Leonard Bernstein. To achieve great things, two things are needed: a plan and not quite enough time. That's where we are. Thank you all very much.
All right, I think we've gotten our meeting off to a fabulous start. I want to thank all of this morning's speakers for a very stimulating time.